Hi everyone, this is Adil. Let's talk about the basics of pigmentary disorders. The pigmentary disorders can be due to defect of melanocytes, melanosomes, or melanogenesis. The defects of melanocytes include vitiligo, Parabolism, and Lentigenes. And the defect of melanosomes is nevus acromicus, which is also known as nevus depigmentosus. And the defects in melanogenesis are melasma, freckles, albinism. Right, let's continue with vitiligo. The vitiligo is due to absence of melanocytes which leads to formation of depigmented lesions which are also called white lesions. Yes, you can see here. And this is an autoimmune disease in which the antibodies are against the melanocytes and the melanocytes are destroyed. Right, let's continue with Parabolism. The Parabolism is an autosomal dominant genetic disorder. It is due to absence of melanocytes. Absence of melanocytes lead to formation of depigmented lesions or white lesions. This parabolism is characterized by localized patch over forehead, as you can see here. Another point I want to mention here, let me zoom this. As you can see here, this whole part is depigmented area, but in between depigmented area some pigmentations are seen. Right, now let's continue with lentigens. The lentigens are increased localized number of melanocytes. This increased number of melanocytes leads to formation of hyperpigmented macules. Just you can see here, these are hyperpigmented macules. These hyperpigmented macules are neither raised nor depressed. Only there is change in the color. There is a question, if these hyperpigmented macules are neither raised nor depressed, then what they are? They are actually flat lesions. They are flat lesions. And these lesions are less than 0.5 centimeters. And these lentigens are associated with the syndrome which is known as Peutz-Jeghers syndrome. This Peutz-Jeghers syndrome is associated with GI polyps. The most common site of GI polyps is jejunum hematomas. This Peutz-Jeghers syndrome is an autosomal dominant condition and it is also associated with lentigens. The lentigens are seen perioral and over buccal mucosa. Let's continue with nevus acromicus, which is also known as nevus depigmentosus. This nevus acromicus is not due to defect in melanocytes, defect in melanosomes, defect in melanogenesis. It is due to defect in transfer of melanosomes from melanocytes to keratinocytes. It is due to defect in transfer of melanosomes. From melanocytes to keratinocytes. You can see here the lesion which is seen in the nevus acromicus, which is a depigmented lesion. Let's continue with nevus anemicus. The nevus anemicus is due to catecholamine induced vasoconstriction. Catecholamine induced vasoconstriction. And whenever there is vasoconstriction, the skin appears pale. Now, in the name of nevus anemicus, there is anemia, anemicus. As we know, in the anemia, the skin appears pale. Same here, you can easily remember now, in nevus anemicus, there is pale skin. Now you can see here in this slide, these lesions are nevus animicus, which are depigmented areas. You can see here the normal skin, these are depigmented lesions which are seen in nevus animicus. Right, Dyscopy. Dyscopy is a test which is generally used to differentiate vascular lesions like nevus animicus from nevus acromicus. In Dyscopy, a glass slide is taken and this glass slide is pressed over a lesion. If you take a normal skin and if you press this normal skin by a slide, this portion of the skin which is pressed by the slide will appear pale, which means this paleness is known as blanching. The blanching will occur, and this portion which is not pressed by the slide will appear normal, and this portion will appear pale. And now we will differentiate the nevus animicus from nevus acromicus. Right, now let's differentiate nevus acromicus from nevus animicus. On Dyscopy, the depigmented lesions of nevus acromicus, this you can see here, these are the depigmented lesions of nevus acromicus. Whenever the glass slide is pressed over the lesion of nevus acromicus, the area which is under the glass slide remains depigmented in case of nevus acromicus. But in nevus animicus, if you take a vasoconstricted skin, this is vasoconstricted skin, and if you take a glass slide and will press over the vasoconstricted skin, this skin will blanch. This area of the skin which is under the glass slide will blanch, and here you can see in this area the blanching is not seen. The area which is depigmented remains depigmented. The blanching is not seen in case of nevus acromicus, but the blanching is seen in case of nevus animicus. There is another way by which we can differentiate nevus acromicus from nevus animicus, that is heat. On heat, the nevus acromicus lesions develop erythema, means the redness of skin occurs due to the vasodilation, and in nevus animicus on exposure of heat the erythema does not develop. Right, now let's continue with defects in melanogenesis. The first one is melasma. The melasma is due to increased melanogenesis. Whenever there is increased melanogenesis, the hyperpigmentation occurs. As you can see here, the dark skin due to the hyperpigmentation, same here. Right, now let's continue with another melanogenesis defect which is known as freckles or affletes. The affletes are due to increase in localized melanogenesis. The hyperpigmented macules are seen, as you can see here in this slide. The hyperpigmented macules are seen just here all over the face, and these lesions develop over sun exposed sites in those individuals who have fair skin. These freckles are seen in normal skin type, that's type 1 and type 2 skin, and these freckles are associated with a disorder which is known as Xeroderma pigmentosa. Xeroderma pigmentosa. It is an autosomal recessive condition in which nucleotide excision defect is seen. Nucleotide excision defect is a DNA repair defect which is seen on exposure of sunlight. In nucleotide excision defect, DNA once damaged do not get repaired. The another feature which is seen in Xeroderma pigmentosa are freckles. These freckles can be seen over dorsum of hand or face, and in young children these freckles can get converted into squamous cell carcinoma. Right. Now let's continue with another melanogenesis disorder, that is albinism. Albinism is an autosomal recessive condition in which tyrosinase gene is defective. When the tyrosinase gene is defective, it means tyrosinase enzyme is also defective, which is responsible for the synthesis of melanin. When the tyrosinase enzyme is defective, it means there is deficiency of melanin synthesis. But remember one thing, there is deficiency of melanin synthesis, but melanocytes are normally present and distributed. Let's have a look on clinical features of albinism. The main clinical features of albinism are generalized depigmentation of skin, hair, eyes. These depigmentations are due to loss of melanin production. And the albinism is also associated with photophobia and nystagmus. Nystagmus are involuntary, rapid, and repetitive eye movements. And the another risk in the albinos which is seen, that is risk of skin malignancies, that is squamous cell carcinoma. As you can see here, the albinism is also known as oculocutaneous disorder. Why? There is a defect of eyes and the defect of skin. That's why it is known as oculocutaneous disorder. Hope you have enjoyed this video. Please subscribe my channel. Thanks.